All right, it's time for our very last lecture video. This is chapter 12. When we voted in class on yesterday, we decided to do chapter 11 in class, and then we would do the lab together in class, which I do believe was the right choice. And I'm hoping that everybody, after you finished your lab, agrees it was the right choice. But now we have this one last little lecture video. Heads up, it's early, as you can tell by the light behind me, and it's cold, and so there's going to be a lot of and possibly quite a bit of cough, because as you know, I'm worse in the morning for this. I have my heater going, so if you hear a weird noise, that's what that is. But I guess that's it. So here we go to our last chapter, chapter 12, DNA technology and genomics. Now this one has a whole lot of added stuff in there because there's a lot that I like to cover, and this version of the textbook, the PowerPoint they provided, highlighted some of it, but I wanted to go a little more into some of it. So there you go. All right, so introduction. You know, for the introductions, they like to give you a little bit of info, and this one's talking about a deadly pathogen that destroyed the papaya industry, basically. Well, threatened the papaya industry. And so this is one of those cases where a genetically modified organism is what was used in order to save it. Yeah, there you go. And so here's where it's just letting you know we're about to talk a lot about GMOs. Again, GMO stands for genetically modified organism, and the various DNA technologies that we use. We talked about cloning different things last time. This time we're going a little more into it and about, again, the GMOs. This is clearly just wanting to point out, are they safe? And it's funny because this is one of the really important topics, because I will go on and on and on and on about this, and when I ask about it on the test, and yes, I do ask about it on the test, somebody will tell me how horrible everything is. So anyway, gene cloning and editing, genetically modified organisms, DNA profiling, and genomics and bioinformatics. Which, by the way, this picture and this picture, I'm pretty sure I deleted from the PowerPoint, because you don't need to see just what the place looks like. And this one, I decided to use something else when we got there. But this is them basically trying to decide what somebody looked like back then. All right, gene cloning and editing. Gene cloning is one of the applications of biotechnology. And again, just break down the words. Bio for life. So this is literally saying life technology. This is where they have the manipulation of different organisms or parts of them in order to make a useful product. When you blend two different things for DNA, that's recombinant. Recombinant literally just means like recombining. So that is where they're mixing multiple parts together, minimum of two. It just means it's not only one thing. And so what it's telling you here is that the researchers can manipulate the bacterial plasmids so that they contain genes from other organisms. So they're taking this one plasmid and then mixing other things into it in order to, I don't know, insert it basically to stuff. So the recombinant bacteria multiply into a clone, because as we talked before, the asexual division would basically give us little clones. And then those genes are also duplicated, and copies of the gene or its protein products can be harvested. This way they can move it along. These are so cute. These are glowing fish, basically. They spliced in the bioluminescent gene from certain jellyfish. This has been done in a couple of different animals for various purposes, and it is normally for research and studying, so you can see like exactly when something develops and whatnot. However, these guys, I remember for a while, long time ago, and by long time ago, I mean like, I don't know, 10, 15 years ago, I knew somebody who purchased the glowing fish, and so it ended up becoming a market. So for these particular fish, I don't know if they actually did any research, or if this was totally just for what it's called industry, just for industry, so just for money. But I'll talk about a different one that glows. So, overview of gene cloning. This is where they're just taking, you've got the E. coli bacterium, the plasmid is that little circle inside of it. I guess I could do this because I have the little laser pointer. This is the plasmid, so they're taking that, and then they're taking the DNA from what we're wanting to clone, they splice it into the plasmid, and then that comes down here, and then it starts, notice recombinant, so again, this is where it's recombined, and then it sits there and multiplies a lot of cells, and then they take those and stick them wherever they want them. So here are various options for what they do, because most people, when you hear GMO, the average person thinks, oh my gosh, people are just messing with our food. And that's one right here. It's like here it shows where they've been working on the corn and stuff like that, because a lot of food product, a lot of produce, they have genetically modified in order to make it a little more resistant to certain insects or weeds. Some of it is more drought resistant, so you can actually grow it in a harsher climate. There's a whole bunch of reasons, but we'll talk about that more later. Another example here is where they have actually modified certain bacteria to consume oil. That way, when there's an oil spill, they can throw the bacteria up there and it munches away and helps clean it up. That's pretty cool. No, it can't clean up an entire massive oil spill. We still need to just stop using the crap. But at least we're coming up with a way to start cleaning up some of the waste. These are the two a lot of people don't think about when they're sitting there bashing GMOs and saying that they're evil and horrible and we need to stop. That's when people say that, it just shows lack of understanding or willful ignorance, and I'm not sure which, but I guess it depends on the person. But here are two huge reasons for GMOs. One, this one, it's showing you insulin. Insulin is one of the original GMOs, because it used to be that in order to, if you were diabetic and your body did not produce insulin and you needed insulin, you had to use insulin that was harvested from a pig. And there are certain cultures where that is a huge no-no. There's a lot of cultures and religions where pork is just, you don't use any kind of pork product. And so say that you're an Orthodox Jew, that's one of the groups that does not use pork at all. If you were an Orthodox Jew and you were diabetic, you would be stuck between having to use something from a pig, which is against your religion, or going into a diabetic coma. Not a good thing to have to choose between. I mean, imagine having to choose between your religious beliefs and your survival. It's really hard. But because of the genetically modified bacteria that science researchers, they worked on it and they made it where these bacteria now produce insulin for people. Now, you can be an Orthodox Jew, still be Orthodox, still be religious, still stick to every aspect of your religious beliefs, and you can still actually use the insulin. So, yay. So that is like one of the best on the GMOs. And then here we have this one where they have actually modified some of the stuff so the protein can dissolve blood clots in the heart. This way, if somebody has had a heart attack and they have clots up there, it's a GMO that they can put in there that help ease it up, so it helps you survive. So GMOs are a lot more than just, food. All right, so in the example shown in figure 12.1b, what is the vector? 12.1b, is that the picture we were just looking at? Yeah, so in this picture right here, they want to know what is the vector. It's the plasmid, isolated from E. coli bacterium. It's what we showed up at the very top. So the vector is basically what's being used to move things along. Restriction enzymes. We talked about this when we did the DNA. Did I call it DNA stuffs or DNA lab stuffs? But I talked about the restriction enzyme. I talked about EcoRI, and the picture on the thing is like GAATTC, and I said that it cuts right between the G and the A, and about how the bigger pieces are heavy, so they stay up here, and the little pieces are lighter, so they go down here. That's what this is talking about. So we've already talked about it. And then we have the DNA ligase will paste the DNA fragments together. And so when it asks what are sticky ends, well, I kind of just told you that right there. It's the single-stranded regions of the DNA fragment whose unpaired bases can hydrogen bond to complementary single-stranded regions of another. So you cut it, and then you can attach it to others. So when I talked about the EcoRI and it cutting it into the tiny pieces, this is how you can take these pieces from this DNA and these pieces from that DNA and hook them together. It's because they're going to match up with each other. So basically making them like little Lego blocks or something. And that's what this is showing you. So see, it's going to show you GAATTC, the exact thing I told you about, and it's got the EcoRI, the exact one I talked about in the little lab, because that's what they use. So it breaks the DNA in that exact spot, and then you attach it. The sticky end is literally where it had already been cut. So then it can place right onto the other one. And then once you have hooked them together, that is now the recombinant DNA. Because again, recombinant just basically means recombined. It's just put back together. So, short single-stranded molecule labeled DNA called a nucleic acid probe can tag a desired nucleotide sequence. Yeah, I don't think I need to reword that one. So, checkpoint question. How does a probe consisting of radioactive DNA enable a researcher to find the bacterial clones carrying a particular gene? Well, it's because when it's labeled, you can find it easily because you know what you're looking for. You've already put it in there and said, hey, this is what I want to find. That glowing jellyfish gene talked about a little while ago. We have glowing pigs that I'll talk about later, and that's pretty much this. It's because then you can find it. You can find exactly where it goes, so you can watch how that part is triggered and how that part works. Pretty sure, yeah. Yesterday I talked about the PAX gene, and I talked about the fruit flies and the eye development, the iris for the human eye. That's the same kind of thing. So when they were trying to find that specific gene, they could attach a probe so then they can find it, so they knew when it developed exactly where it was. Complementary DNA. Well, there you go. It can be used to identify the genes that are being transcribed by a particular cell at a given moment, because complementary just means the one on the other side. So like when the A is here, the T is here. That would be the complementary. Why is the use of a viral enzyme critical to producing complementary DNAs? Well, because it's reverse transcriptase produces DNA from RNA, and most cells lack this enzyme, so it just kind of gets it going. CRISPR-Cas9 system allows researchers to target a specific gene in a living cell for removal or editing. CRISPR is actually the machine, basically, that's used. You'll see it. Well, I can't say you'll see it. If you watch crime shows, documentaries, and a lot of sci-fi, they'll make a comment, and you'll just, if you pay attention to the background, you'll hear them say something about the CRISPR, and that's what this is. And so what it does is it clones exactly the copies of the gene, and then the CRISPR-Cas19 system will modify the gene. So this is how they're doing like the gene therapy and I guess the GMOs and stuff. So this is just showing you how the whole thing works. We've got the cell. This whole thing is a cell. Cytoplasm, so that's the liquidy part of the cell. Here is presumably the nucleus of the cell. And so Here's the protein. We've got the guide RNA right here. It comes in here, and then that kind of like poof goes in. And then that's how this DNA starts to get rewritten. It writes it in there. And then you end up having your new stuff, your new info. Now, GMOs, genetically modified organisms. And I keep saying it that way because on the test, when I say, what is a GMO? I mean literally, genetically modified organisms. Organisms. That's the important word to remember as well, because I get people who, they come up with all different things for the O to stand for, but it stands for organism. All right, here's a bunch of extra stuff that I added in here. Actually, I don't know if it's a whole bunch right here or later, but I've got a bunch. So anyway, genetic engineering. This is where in the laboratory they deliberately make changes into an individual's genome. And when I say individual's genome, that does not necessarily mean human. Here, we're talking mostly about bacteria and yeast. And this stuff is most often used in research, medicine, and industry. A moment ago, I talked about the human insulin, where they modified the bacteria to make the insulin. So that would be a case of genetic engineering. I guess technically that falls under all three categories because it's definitely medicine. They did it in order, they started as research, then it became the medicine, but we're in the US, all medicine is also industry. So they just, money, money, money. And now I'm thinking of Pete's Dragon when the family sings money, money, money by the pound. Anyway, genetically modified organisms. Yes, I'm going to say it a couple of times because you need to remember that's what GMO stands for. So a genetically modified organism is simply an individual that contains modified genes. What I think is interesting is if you notice here, it says from the same species or a different species. Sometimes it's just its own genes that were tweaked. Other times they add something in from somewhere else. Then we have transgenic organisms. These are individuals that contain genes transferred from a different species. These are also GMOs. So transgenic means that it has genes from somewhere else. That's what the trans stands for, is from outside. Example, bacteria with jellyfish genes. I used to have a picture of the bacteria with jellyfish genes. I guess I didn't include it here. Oops. So bacteria, yeast, cell cultures, and whole animals can be genetically modified to make products for medical and other use. Yeah, pretty much. They do this in order for various things. Like before, I told you the bacteria for the human insulin. Just a moment ago, I told you about using the jellyfish genes in the bacteria. That was in order for it to glow so you could watch different stages occur. And we also talked about the fish, the cichlids, a moment ago that glowed. And so there you go. Now, why can't all human proteins be synthesized in E. coli, like in the example of the plasmid? Because the bacteria cannot correctly produce some proteins, such as the ones that require the attachment of sugar groups. That's why we can't use human proteins, can't have them all synthesized in E. coli. Now this one is in your current text, where it talks about the goat carrying a gene for human blood protein that's secreted in the milk. This one I had to look it up because I knew that it had the human blood protein, but I couldn't figure out exactly why. I had heard previously just human protein, and I assumed it was for like baby formulas, milk, and stuff like that for infants, because they have made that before. But this one, when I finally noticed it said human blood protein, I was like, so I actually researched this one. Not sure if the textbook goes into depth on it, but just in case you were curious why they would do this. It's because sometimes people need more of the clotting factor that's found in plasma. We don't have a whole lot of people who are donating their plasma or selling their plasma, because I'm pretty sure this class we had to talk about the plasma donation, but that might have been 124. Anyway, this way, they do this, they milk the goat every day because that's the way it works, and then they can actually separate it and they pull out the human blood protein that can then be used for human issues where they're like lacking the clotting and stuff. So, interesting. All right, DNA technology has changed the pharmaceutical industry and medicine. Well, we already knew that because I've already talked about the insulin a couple of times. But also going to point out that this can also produce drugs like the insulin, diagnose diseases. That's important. That goes with the genetic stuff when we were talking about colon cancer and stuff. And produce vaccines, harmless variants, mutants, or derivatives of a pathogen. Producing vaccines, that's a big one. So a lot of vaccines, they use a lot of times various bacteria, yeast, and a lot of times a chicken egg in order to produce or develop or fine-tune the vaccines, all of which have to do with genetically modified stuff. So, checkpoint question. If insulin and human growth hormone are both natural products, why use genetic engineering to make them? I think we already know this answer, right? Because I've already said it. Because we can provide them in much larger quantities than you can obtain naturally. And it also helps with the ethical issues of various cultures and religions. That if you can't use, say, again, pig, then this is a better way to go. Scientists have produced many different varieties of genetically modified organisms. The genes transplanted from one organism into another, typically of another species. The recombinant organism is called a transgenic organism, which we already talked about a moment ago. And a whole bunch of important crop plants are currently genetically modified. So what they do here is, again, they take the bacterium, they take the plasmid. Here they say TI plasmid, and that's for tumor-inducing, just so you know. And tumor-inducing does not mean giving you cancer. It just means it makes that little tumor that stays in place. So think benign if you need to. Okay, so then they splice the DNA in there. We've got the TI plasmid that now has the DNA of what they want in there, and then it goes into here the plant, and then that gets added into the gene, and so the chromosome now is part of it. And now as you grow the plant cell, you've got the whole new plant that now has that trait that you wanted it to have. Which brings us to golden rice. Golden rice, I'm pretty sure I made reference to this in class already, and this is where they have spliced in vitamin A. This way, in my brain blanked on the word. I'll just say areas because I can't remember the word that started to come out. In areas, locations, whatever, where there is a lack of nutrition, but they can grow ample amounts of rice by giving them the golden rice to grow and harvest the plants, then all of those people in that area can now consume this rice instead of plain rice, and they're all taking in some much needed vitamin A. The first time I read about any of this was my first semester in college, and it was spinach, where they had spliced in protein into spinach so impoverished nations could get a higher amount of protein from spinach, which is what they grew. And the only reason I really remember this is, for some reason, my very first semester, while I was taking remedial math and introductory courses, for some reason I decided to take intro to microbiology because it just sounded interesting. And my teacher, she was very entertaining, but she also had cancer, and she was in the later stages of it. She actually passed away that summer while I was doing my first my first study abroad in Belize. And so they let me know because everybody knew I liked her. And she actually didn't even make it all the way to the end of our semester. So I had her last semester. But anyway, she's the one who encouraged me to just read all the different things. And I went and I read it. And I read about this, the adding protein to spinach. And I remember going to her office and telling her about it because I thought it was so fascinating. And what I never forgot was her looking at me and saying, if only they would actually put it in food people would eat. And I was like, what? Because I like spinach. But it just cracked me up. And so I honestly couldn't forget that. She was pretty cool. All right. Use of genetically modified organisms raises questions and concerns. So scientists are investigating the potential risks to human and environmental health posed by the DNA technologies. As you know, a lot of people will tell you how horrible GMOs are. I'm hoping I have shown you that there are actually some benefits to GMOs because we've talked about a few and we're going to keep talking about it. But why is it often necessary to run both human and animal studies to learn about human health? It's a good one to think about. It really is. Because sometimes when you hear about the studies, people are like, oh, it's horrible to do any kind of animal testing. But in reality, we kind of need to study both, and animal testing and animal studies don't necessarily always mean cruelty, just so you know. So here, animal diets and lifestyles can be very closely controlled. Humans, not so much. You can tell a group of humans that we're trying to see how this affects you, and we need you to stay to this strict diet and do these specific activities, and you will have some who will do exactly what you ask. You will have others who deviate just a little bit, and others who will just straight up lie about whether or not they did it. Animals, you're kind of in control of every aspect. If you tried to put humans in individual rooms and control every aspect of their lives, people would pitch a fit and say, oh my gosh, that's cruel. So that's kind of why you have to do this sometimes, because if you need to know if this one product is causing an issue, you need to make sure that that is what you are studying. Okay, so here, vitamin A. Of course, we're doing vitamin A since we were talking about golden rice. So vitamin A production after consumption of different sources of beta-carotene. So if you take a capsule of straight up beta-carotene, then it shows here that you have 50% absorbed and converted to vitamin A. If you eat the golden rice, which is what they made, notice where that bar graph goes. That would be what, right around 40, about 40%. Spinach is, what is that, about 15%. So there is vitamin A in spinach, but it does not actually convert a whole lot of it to pure vitamin A in your body, but the golden rice does. And yes, the capsule of pure beta-carotene does the most, but you can't exactly constantly give a capsule to everybody in a nation. But you can help them with the rice. Now we have gene therapy. We talked some about this yesterday as well when I was talking about the colon cancer and stuff like that. So gene therapy is changing a defective gene to a normal one in a living human, and this shows promise for curing defective genes, but the actual successes are rare. So we're seeing the beginnings, but basically we don't know. We haven't fine-tuned it, I guess is the best way to put it. So checkpoint question, why does bone marrow make a good target for gene therapy? Well, because that's where everything's made. So the bone marrow cells multiply throughout a person's life and contain stem cells, like we talked about yesterday, that give rise to different kinds of blood cells. We talked a little bit about the fact that your bone makes the blood, and so there you go. All right, we talked about colon cancer, and here we're going to the different types of therapy. So we have a healthy person. We've got their gene, a nice normal gene, normal allele. Then you take the RNA genome of the virus, the retrovirus, put it in there. The bone marrow cells are now infected with the virus. Notice virus does not always mean detrimental. I have to specify that because when you say virus right off the bat, especially now, you're going to think things like coronavirus, and you're going to think of things that'll make you sick. Not always the same, but here we go. The bone marrow cells are now infected with the virus, so the bone marrow cell of the patient, we take the viral DNA carrying the human gene, inserts into the cell's chromosome, and then the engineered cells are injected into the patient. So it's basically getting everything to work together to create it all. I was going to say, here we go again, but no, we've already done this slide, genetic engineering. Apparently, I accidentally have it in here twice. We've already talked about this part. There's the picture I thought I had. See, I wasn't crazy. I did have the picture. I just accidentally put things in there twice. So here's the bacteria with the jellyfish gene. This way they can actually see when certain stages are happening, where certain things are, that kind of thing. Now we have designer plants. Designer plants meaning things that have been genetically modified to be very specific, and these are all over the US. The Thai plasmid we were talking about a moment ago. Thai, TI, stands for tumor inducing, and that means it's making the place to put the genes. It's literally where it's going to get placed in order to modify what we're modifying. I spoke a moment ago about this. Here it's just to help you remember about the crop plants that are being genetically modified. It's to produce more food at a lower cost. It's so more people can be fed for less. That kind of thing. Resistance to disease or herbicides, this way they can survive longer. Increased yield means to produce more food. So imagine a corn plant, a nice tall thing of corn, and imagine more ears of corn on that corn plant, or more lettuce leaves, more spinach leaves, that kind of thing. They're trying to make more come out of the one plant. Plants that make pesticides, it's a Bt protein. I used to give you an online lab about this. I don't know why I didn't do that, unless that's this week's. No, we did our genetics lab. So I don't know if I give you this lab unless it's for next week. But I normally do one for the Bt protein, and it shows that it helps keep the insects from munching on the plant. And then drought resistant. I mentioned that before because if you're paying attention to the world, you will notice that we are having more and more droughts, more and more severe droughts. And it's important for our plants not to immediately die when we have a longer dry period, because if all of our plants keep dying, the drier it gets, we're all going to die. I mean, that's just all there is to it, because we have sucked so much of the water out and wasted so much, and yeah, we kind of need to make sure that there's a way for our food crops basically to survive our mismanagement of our water. So 73 GMO crops are approved for use in the US, with hundreds more pending. So what I like to point out here are some of the GMO crops. We've got corn, sorghum, cotton, soy, canola, and alfalfa. Plenty more as well. What I like to point out here is I know people who will sit there and tell you how bad GMOs are, how they refuse to eat it, and that's why they only eat, and then they'll list some stuff, but more often than not, they will mention some kind of meat, at which point I will point out that their meat is actually eating stuff that already are considered GMOs, because if you eat cow, the cow eats corn and alfalfa. If you eat chicken, they pretty much also eat the corn and the alfalfa. If you eat pig, same thing. So pretty much if you're eating meat, your meat is probably already eating the GMO crops. So you can't sit there and be all, oh, I just don't touch that stuff if you eat any of the other stuff. So, but again, it's not that it's bad for you, it's continuing. One of the biggest things, the issue I have, is when people tell me that if you eat a GMO crop, it's going to change your DNA. At which point I ask them, when you eat an apple, do you become an apple? And then they laugh at me and say no. But it's the same logic of saying if you eat a GMO crop, it's going to change your DNA. That's not how it works. That's not how your DNA works. So just chill. But I am going to get to some downfalls in just a moment. Facts and controversy. In crops engineered for herbicide resistance, weeds are becoming resistant to herbicides. That's true. And engineered genes are spreading into wild plants and non-engineered crops. That is also true. So those can be very big negatives right there, like huge, because you also want the pure crops. But yeah, I mean, one of the ones I consider as being one of the biggest dangers is, I've talked before that I'm highly allergic to mushrooms. Mushrooms, very good for you. They're very healthy. They're the protein and the high vitamin D. There's great reasons to use mushrooms in just about everything, unless you're allergic to mushrooms. So my issue is if they decide to modify some other kind of vegetable that I eat, splice it with genes from the mushroom, I am not guaranteed that I'm not going to get a reaction. I don't know what part of the mushroom gives me the reaction. I just know all mushrooms, and blue cheese and penicillin and things on those lines all actually affect me. By the way, the blue cheese is because blue cheese is made with a type of penicillium, which is related to penicillin. So if you know anyone who's allergic to penicillin, they probably should steer clear of blue cheese, because they might not be massively allergic to it, but they might notice that they get some kind of odd reaction every time they have blue cheese. Just handy tip. But anyway, with those types of allergies, if you have various food allergies, if you're not aware of what was spliced into the other food crop or the food, you may or may not get an allergic reaction to something you didn't think you were allergic to. Now, is this a common occurrence? Don't think so. But when you are discussing possible issues, that's one that you really need to take into account. So maybe if they at least list what was used in each thing, so then you can make a judgment call. I think that would be a fair thing. Biotech Barnyards. I always mention this. I find this interesting, and I always ask about this on the test. And I will not say it on the study guide, because I expect you to have, actually done the video. It's awkward to sit here and make the videos, especially when I'm really cold and I'm shaking, trying to keep warm. That brings up that whole negative feedback thing I think I mentioned in this class about what your body does when you're cold. But sometimes it's stuff just what I say in class or in one of these videos to make sure that you're doing your part, because I don't make these videos just for myself. That's for darn sure. It's very awkward to sit here staring at this green dot and talking. But anyway, Biotech Barnyards, biotechnology. That's what biotech stands for. Barnyards, we're talking animals. So these are animals that would be impossible to produce by traditional breeding methods. So they have to use genetic engineering. These animals are used in research, medicine, and industry. You will hear that all the time. Research, medicine, and industry. So here's the first one, 1982. They took mice, rat genes, spliced them into mice. So they took the genes for the rat growth hormone and spliced it into a mouse to see if the mice would then grow larger, basically, or to see if they would be able to utilize the rat growth hormone. So, other examples. Well, they used the modified animals to figure out the human diseases. I spoke yesterday about something called knockout experiments, and again, that's where I talked about the fly and the eye development and the PAX gene and the iris. And yes, I kept doing this every time I talked about the iris for the eye, for the human eyes. So mice are used in the knockout experiments often so they can see exactly where that gene is. They normally start with something a lot more simple, again, like a fly, a fruit fly, because they reproduce so rapidly. Eventually, you have to move up. I was about to say up the food chain. That just sounds wrong. But you have to move up the chain of advancement. I can't think of the right word, but anyway, a mouse is at least a mammal, and so that gets closer to what humans are. So you kind of have to move up the chain in order to make sure things develop similarly. All right, so genetically modified animals make proteins with medical and industrial applications. So again, goats and rabbits that make human proteins. We already talked about the goat and the human blood protein. And then farm animals with desirable characteristics. So here you go. I always talk about these three Biotech Barnyard guys. So right here we have goats. See this weird little thing here? These guys have been modified to produce spider silk. Spider silk has a very high tensile strength, and this is so they can use this to make various things. I think the most interesting ones I read about was like for bulletproof vests and stuff like that, or really strong cord or something like that. But I think anyone who's ever accidentally walked through a spider web knows that that sucker is a lot stronger than you would think. And being able to use that to make other things makes sense. However, how many spiders would you need to get enough of that silk to do what you want to do? So they modified the goats. So every time they milk the goats, they've got the spider silk. These are the glow-in-the-dark pigs. You can tell this guy looks a lot different than this guy. So the glow-in-the-dark pigs, it's not just because, oh look, hey, this sucker glows. It's because when they are developing as embryos, very similar to human embryos. And so by having certain parts of them glow, you can actually see when each of the different organs are developing, and you can try to find when certain abnormalities take place. That way they can learn better how to treat babies as they're developing if something goes wrong. So there you go. But this dude, man, this is the main one. You have to know there's something wrong with this chicken. The number one thing is he has no feathers. Or I should say she has no feathers, but I don't know. It could be a rooster. I have no idea. But this chicken has no feathers. Well, it's because this one is a perfect example of industry. Somebody realized that by having chickens with no feathers, it would cut the processing time down by a couple of seconds per chicken. I think it was 15 seconds per chicken, because you don't have to pluck them after you kill them in order to process them for their meat. So they got rid of all the feathers. But also by getting rid of the feathers, you didn't have to have the same kind of air conditioning in the areas because they didn't get overheated as much. Then it turned out that they were actually a leaner meat, because by not needing the extra fat to help hold the feathers in place, it got rid of a little bit of the fat. So it ended up being a lot more, money-wise for the industry for people who eat chicken. The issue, however, is the way that birds reproduce is the male kind of flutters behind the female. And without feathers, they can't flutter. And so these guys have to be artificially inseminated in order to make new babies. There you go. Those are your Biotech Barnyard animals. This I totally kept just because I love the word xenotransplantation and I wanted to make sure to still get to use it. So, transgenic pigs with human proteins are a potential source of organs and tissues for transplants in humans, and that they may prevent rejection by the immune system. By being able to modify these, being able to use their organs in humans is huge, because there's not enough organ donation going on to help people who need new organs. So xenotransplantation is just the transplantation of a tissue or organ from one species to another. So xeno is other. That's why anyone who watches sci-fi, if you ever watched an alien movie or anything and they talk about a xenomorph, that just means a different body. So safety issues, I already talked about some of this. I talked about like the possible allergens and stuff, but here's a different one. first transfer of foreign DNA into bacteria ignited an ongoing debate about potential dangers of transgenic organisms that may enter the environment. So, early questions. Could genetic recombination in laboratories produce a new superpathogen? Yes. Yes, it can. What if new forms escaped into the environment and transformed other organisms? It's already happened. We've already talked about that. There are a lot of plants that were not originally modified, but because of the way pollination works, they have now been modified. There have been other cases. If you just, Google some of this, you will see a lot of interesting cases, because things have a way of not staying as contained as people might want them to. So, safety guidelines for transgenic research were adopted in the US and other countries. So basically they said, hey, you just can't go this far. Which brings us to modified humans. As a society, we continue to work our way through the ethical implications of applying new DNA technologies. Manipulation of an individual genome continues even as we are weighing the risks and benefits of this research. So we're still seeing if we can do it while we're trying to decide if it's a good idea. And this is one of those things. I used to say this all the time, and Jeff Goldblum, I believe, says it in Jurassic Park, and I still totally 100% agree that too many people have a tendency to do that, can we do this? And forget to stop and say, should we do this? There's a lot of good, but there's a lot of crazy that can happen too. And so having particular regulations in place, I would say, is probably the best way to go. Because now we're going to talk about gene therapy. We've talked about some of this where it pertains to cancer. Here's another one. So, transfer of recombinant DNA into body cells to correct a genetic defect or treat a disease. We have the viral vectors or the lipid clusters that will insert the unmutated gene into the individual's chromosome. The unmutated gene means the healthy, normal gene, not the diseased one. So this is basically trying to take a healthy gene and replace the one that's not working properly in the person's body. Examples of this would be like cystic fibrosis or SCID-X1. SCID-X1 is the one we are about to talk about. So, 1998, they used a viral vector to insert the unmutated IL-2RG gene into boys who had this disease. SCID-X1 stands for Severe Combined Immunodeficiency Disease. Most of the boys who had this gene inserted recovered immune function. So they ended up being perfectly healthy, as in they got rid of their disease. However, nobody can predict where the virus injected gene will insert into the chromosome. That part we can't control. Several boys from this study developed cancer due to having this take place. In other studies, some of them had a severe allergic reaction to the actual viral vector itself, and then they died from the severe allergic reaction. So you have a group of little boys where most of them ended up becoming healed, like healed, 100% healed, never have to deal with it again. You had some of them develop cancer, and a smaller amount of them died. That's where you run into the whole ethical dilemma. Because since you can't predict where it's going to go, you can't predict how many or who's going to get cancer. If you don't know about the possible allergen for the individual, you can't control that aspect either. But you have to decide, is the healing of the majority more important than the cancer and the deaths of the minority? And that's where it gets really difficult, because it depends on who you ask. But then again, that's ethics. That's part of why I like ethics. Makes you have to think. And then that gets us to the eugenic engineering. This is where they're trying to engineer humans for a particular desirable trait, not having anything to do with treating a disorder. This would be like deciding, I want my child to be born and be super athletic, or I want a kid with I don't know, blonde hair and blue eyes, or I want my child to be 6 foot tall and easier time becoming muscular, stuff like that. So it's basically, if you've ever heard the phrase designer baby, that's what this is referring to. I have all kinds of issues with this because I don't think anybody knows what is actually important. And sometimes I think nature gives you what you get because you need to learn how to, you know, let other people be, basically. And perfect for you is not perfect for everybody, and that gets very dangerous very quickly. Not to mention, if you're deciding you want to make sure this gene is in there, what gene are you getting rid of? And is that then the individual's choice anymore? You know, there's so many ways to go with this. I've had a lot of ethical talks about this one. So that leads to here. Eugenics, selection of human being with the most desirable traits. Eugenics is what's been used for the excuse for a whole lot of things of genocide. Entire areas where an entire group of people is destroyed because someone else decides this is the better human. And so we need to get rid of all the non-perfect humans. But again, there is no perfect. And the world is so much more interesting having a wide variety of people. So it's just, I don't know. I find it disgusting, personally. I don't think that you should engineer humans for any particular desirable trait because you don't know what you're getting rid of in the process. What if you decided that the child you want is the perfect athlete, and so you have the genes tweaked to make them a perfect athlete, and in the process you destroy whatever curiosity or intelligence the kid would have had where they could have solved some world problems because you wanted someone who could play football better. You know, I'm not downing football, but not knowing what you're giving up to get what you want, you're going to miss something important. Or at least that's the way I see it. So that's something to think about because that is something they've been working on. So the question is, would it be okay to genetically engineer superhumans? And yeah, the military has been checking into this in order to make the perfect soldier, which again is disturbing because if you design the perfect soldier, a perfect soldier also follows orders, which means you have designed someone specifically so they can fight for you and do things for you, but don't really have much choice in whether or not they follow the order, at which point we're kind of towing the line on taking away the free will. But again, that's my whole philosophical and ethical conversation there. Using the new technologies, they really are checking into these things, and like I said, it raises more and more ethical questions. You have to start really thinking it through before you say it's good or bad, because there's going to be cases for both. And that brings us to DNA profiling. That means literally getting the lineup of your DNA. So this is used a lot in forensics. We talked again in lab about like the crime scene DNA stuff or paternity tests. And so that is going to be with the DNA profiling. Crime scene, similar to what we did in lab, where we've got the DNA, it's been isolated, we run the gel and we see what's been left at the crime scene, and then we compare it to the different suspects. So here, according to the data presented, which suspect left DNA at the crime scene? There you go. We can see that it's suspect 2. So again, just like we did in lab, it's the entire profile that you're comparing. The way we get this is through a PCR, polymerase chain reaction. It's a machine. It like goes when I do that, it's like it heats up, cools down, heats up, cools down. It causes it to keep replicating so we can make a whole lot of copies of one little sample. That way we don't lose the whole sample after we run one test. I did some of this in grad school. I wanted it to go well because it fascinates me, but I discovered I suck at this. It's just not my cup of tea. I had a whole image in my head of I then run it through the machine and then I do the little things. I've got these beautiful gels. No, I sucked straight up. It was bad. But polymerase chain reaction, PCR, that's how they amplify the DNA sample. By amplify, again, it just means they make a whole bunch of copies. So then you can do more and more studies and research. This is how it works. You've got your sample DNA and then heat. That's why I said heats it up, cools down, heats it up, cools down. So heat separates the DNA strands, the primers bond with the ends of the target sequence, the DNA polymerase adds the nucleotides, and then it happens, and it happens, and it happens. It goes through multiple cycles. Each cycle, it makes another strand of DNA. Because if you remember, I said in class that DNA replicates from a semi-conservative replication. So what happens is it breaks and you have 3', 5'. And I'm pointing this out for a reason, because it will always replicate from 5' to 3'. So it starts here, continues there. So it starts on the 3' end and it grows from 5' to 3', 5' to 3'. Down here, notice 3's here, but it's 5' to 3'. So it goes from the 5' to the 3' direction. They always, always, always go the same direction like that. It's going to be the opposite. And that's because if this is a G here, then you're going to have a C here. So it has to go the opposite direction. But anyway, each cycle, it spreads it apart again. And so it builds yet another sample underneath it. That's the semi-conservative replication. This is the old strand, this is the new strand. So there you go. Why does PCR amplify only one specific region of DNA rather than all of it? It's because the primers are marking the ends, and then that's what's telling it this is where you go. Pretty much. Many DNA technology applications rely on gel electrophoresis. This is what I talked about in class with the gel. And again, the skipping the rocks. The heavy piece stays up here. The tiny piece goes all the way down. That's what this is telling you. So it's separating the things based on size and electrical charge, different things like that. So what causes DNA molecules to move towards the positive pole during electrophoresis? And why do large molecules move more slowly than smaller ones? Negatively charged phosphate groups of the DNA are attracted to the positive pole. Same thing as magnets. If you remember chapter 2 when we talked about polar versus non-polar, and I talked about magnets like this or like that, that's what this is talking about. And why do large molecules move more slowly than smaller ones? Same thing. Longer fragments are more restricted by the tangle of fibers in the gel than are the shorter fragments. So in other words, they hang back. They're heavier, harder to move. That's it. So this is what I was talking about, where you put the stuff in and then it pulls the strands down, and that's what separates them. And then this is an actual gel that they have lit up so you can see it. I have another picture too. This is an actual gel, a forensics case. They have, this is size reference. Notice they have size reference in multiple places. This is so you can see what's the smallest and what's the largest. Then they have control DNA. Control DNA is extremely important because it's making sure that you know what it looks like. So you know for sure the test is working. Now here, this is the victim. You have a couple of spots here. You have suspect 1. You have suspect 2. Female cells. Notice the female cells match up with the victim. So you know that's the same person. Then you have the semen, and then you have the boyfriend. So presumably this is a sexual assault case because semen. Boyfriend does not match anything else. Suspect 1 matches the semen. So does this say that suspect 1 is guilty of a crime? Well, like we talked about in class, If suspect 1 says he has never met this female, yeah, it's going to look really bad for him. But if suspect 1 says, oh yeah, well, we were having an affair, her boyfriend just doesn't know, that changes the whole story. Does that mean he's innocent? No. But this just proves that suspect 1's semen was there, and suspect 1 has a lot to explain. So there you go. Repetitive DNA consists of nucleotide sequences that are present in multiple copies in the genome. Much of the DNA that lies between genes in humans is of this type. So it's just repeat, 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 which comes to these short tandem repeats, which I talked about yesterday. Short nucleotide sequences that are repeated many times in a row. And DNA profiling by the STR analysis involves amplifying 13 of these. So here you go. We've got the crime scene. They take that short tandem repeat and then they keep doing it to see whether or not it matches. So we have this one where this one matches, this one does not match, because see, that's perfect match. Not so perfect. So there you go. Again, showing you the way the gel works, where they put it in there and where they land. Applications of DNA profiling. We talked about this yesterday as well. Helps solve crimes, do the paternity test, identifies, that kind of thing. So in what way is DNA profiling valuable for determining innocence as well as guilt? Because you can't prove definitively guilty or innocent with the DNA. However, if there is DNA present, you're accusing this person of having been there, but the DNA that's there does not match this person, that might help prove this person did not do it. It doesn't prove that they didn't do something, but it proves somebody else was there. And there are a lot of people who went to jail many, many, many years ago for crimes that people swore up and down that they were convinced these people did, but when DNA that was held onto decades ago was reprocessed, the person in jail was found not guilty and they were released. I mean, yes, their life sucked for a couple of decades because they were stuck in jail, but at least they were finally proven that, hey, look, that proves it wasn't me. So DNA profiling, extremely, extremely important. That's talking about one of the things I was just talking about, where they show, hey, look, it's not me. And then the person ends up getting released. So there you go. Genomics and bioinformatics, and we're almost done. Small segments of DNA can be sequenced directly. So next and third generation sequencing machines can quickly determine the sequence of relatively short stretches of DNA. So now it's happening a lot faster. And it's because it works on a single long DNA molecule for the third generation. And next generation sequencing involves piecing together many smaller molecules. Genomics is the study of complete sets of genes. This is getting them to understand more and more and more what all's involved. So, does a larger genome always correlate with more genes? Well, here you go. Number of genes is our y-axis. Size of the haploid genome is the x-axis. So here we have tiny. Here we have more number of genes, and here we have larger. So this is where Homo sapiens are, right here. Chimpanzee actually has a few more genes than us. The rat has more genes than us, which you have to make kind of interesting. So does a larger genome always correlate with more genes? Nope. Because look at how many genes rice has versus how many genes a rat has. The rat is larger than the rice, but the rice has more genes. So kind of interesting. Human Genome Project, massive publicly funded scientific endeavor to determine the nucleotide sequence of all DNA in the human genome and identify the location and sequence of every gene. This is so we could understand what everything does. Data from the Human Genome Project revealed that the human genome contains just under 21,000 genes and a huge amount of non-coding DNA, most of which consists of repetitive nucleotide sequences. Some people refer to it as junk DNA, but I refuse that, because just because we don't know what it is doesn't mean that it's not there for a reason. So this is just showing you the human genome and the amount of, I guess, info for the different parts. We talked about introns and stuff like that previously. The exons, that's there, that's for the coding. So there you go. Whole genome shotgun method of sequencing a genome can provide a wealth of data quickly. So pretty much this just means taking a look at the entire genome helps, I don't know, answer a bunch of stuff. So what are the primary advantages of the whole genome shotgun method over previous methods? Faster and cheaper. We're America. We do like faster and cheaper, I will admit. Bioinformatics. The use of computational methods to analyze biological data can be used to analyze large sets of data about DNA sequences and proteins. Proteomics, I think that's how you would say it, involves similar systematic studies of the full protein sets encoded by genomes. So why is studying sets of proteins rather than sets of genes beneficial to understanding how cells work? Well, because it's the proteins that carry out the actual work. I'm hoping throughout this quarter it's been drilled into your head the importance of the proteins for the body. So there you go. I used to have a lab on this, and I need to find it so I can reincorporate it into Bio 124. But this is showing you the sequence, and I just totally got distracted because I realized that says cat. Anyway, you're looking at these different species, 5 species of sea slug. All of them have the same genus. The species is different. These 4 have this little gap, but this one, that gap is filled. So that's what they're trying to show you here. Unlike databases like GenBank, researchers can now compare genome sequences from many species, allowing hypotheses about evolutionary relationships between those species to be tested. Comparison between humans and our nearest evolutionary relatives provides insight into human evolution, which we will cover in Bio 124. So, how can cross-species comparisons of the nucleotide sequences of a gene provide insight into evolution? Well, because it shows as it started to slowly veer. By looking at the DNA, you can see which species are more closely related to other species. And again, we'll cover that in Bio 124. And there we have it. We got it done, and bam, that's all there is. We are finished with what we cover in the textbook. So now you just have to make sure you actually paid attention to this. There are a couple of things mentioned throughout the video that will be on the test, and now you just need to prep for the test. Because on Monday in class, you will have test number 3. After you take the test, I will be grading them. You'll take a break while I grade, and then I will hand them back. It would be in your best interest to bring your previous tests with you to class on Monday, because after you take the test, after the break, when I hand back your tests, you will then design the final. I will be writing on the board some rules, because I tell you stuff that you have to have, and you get to design the rest. Most students actually enjoy this part, but you will have to also go by the rules of making the test. But anyway, Test 3 is in class on Monday, and you're going to have to remember some of the stuff from this PowerPoint. You are going to have to practice your genetics because you will have to answer some genetics questions, do some Punnett squares, and you for sure 100% are going to have to also draw and label meiosis. And meiosis means meiosis 1 and meiosis 2. So those are some definites that will be coming on that test. I have not made it yet, but I can promise you those things will most definitely be on there. So a quick review from the stuff we've covered since the last test would probably be a good idea. And don't forget the scientific method, because I told you that would be on every test and I wasn't lying. So that's it. I'm done with videos for now, so peace out.